Hello, it's Dr. Williams again. We've got another video for you about Biology Paper 1. This time it's the triple only content. There's not a lot of it on Biology Paper 1, but what there is, you need to know pretty well if you're doing this paper. Right, let's go. So, the first thing that's on there is monoclonal antibodies. This comes up quite a lot on any biology papers, so make sure you know what they are and what they're used for, as well as how they are produced. So monoclonal antibodies are made from single clones of cells. Mono meaning one, clonal meaning clone. This means the antibodies are specific to just one binding site on one protein antigen. Now, that doesn't have to be an antigen of a pathogen. It could be an antigen of anything, but it has to be made of protein because that's what they are looking to bind to. So this means they're able to target a specific chemical or specific cells in the body. So they're really useful for a lot of analysis. The way you make a monoclonal antibody, follow this method. So first of all, you inject a mouse with the antigen. You can't feed the mouse. It does have to be injected in their bloodstream so that you make their white blood cells make your antibody in their blood. This means the mouse lymphocytes, and you do have to use that term, not white blood cells, the lymphocytes will make a specific antibody complementary to the antigen. But they will also have other antibodies in there that you need to remove. So what you do is you extract all of the mouse lymphocytes, and then you need to identify the lymphocyte which has the specific antibody to the antigen. So for example, that could be done by placing your antigen in a dish and you add your lymphocytes to it, and those with the specific antibody would attach to it, and they're the ones that you would keep. Once you've identified the lymphocyte with the specific antibody, you would combine it with a myeloma cell. And this creates a hybridoma cell. All of the keywords in bold, it is essential that you use if you're asked to describe this process. So a myeloma cell, or you can use the words tumor cell, actually grow rapidly, and the lymphocyte making the white blood cells will make the antibodies. So when you make a hybrid of these two types of cells called a hybridoma, you're going to get rapid growing cells that can produce lots of antibodies. So at step 5, this hybridoma cell will clone itself to form more identical hybridoma cells, each producing the monoclonal antibody, that same type of antibody that you've identified that's complementary to the antigen. You then collect and purify the antibody produced, and you've got your source of monoclonal antibodies. And the good thing is you can keep those hybridoma cells alive, so you can have a constant source of this antibody whenever you need it. And the reason you might want to do this is for all of the different uses. So you can use monoclonal antibodies in pregnancy test kits. So you would urinate on the bottom of the stick, and you would have one antibody that binds to the pregnancy hormone in the urine. And then this antibody in the antigen will diffuse up the stick window. And then there's a second antibody that's already there waiting to see if the antigen appears. And that's at the result window. If the first antibody has something like an enzyme attached to it, what you will see is a colour change in the positive result window. You always have a control window as well, just to check that it works. Normally in this control window is a third type of antibody that will just bind to the antibody itself with or without an antigen. So usually with these any sort of questions about this application, you'd be given information like you see in that diagram, where you've got the control window and the result window, and some information about how you would decode what any of that means. You don't have to know the specifics of a pregnancy test, just really the application. Another use is in laboratories. So you can use monoclonal antibodies to measure levels of chemicals, which are in really small amounts, in samples of blood or things like that, or to detect pathogens like HIV and AIDS, as well as herpes and chlamydia, which have specific antigens. So these monoclonal antibodies can be made to the antigen of those specific pathogens. The easiest way to see the attachment of any monoclonal antibodies is to actually attach them to a dye. So these dyes, they might glow fluorescent under UV light, and then you can just put the cell sample under UV light, and if there's glowing there, then there's a monoclonal antibody attached, and you will have that chemical present that you're looking for. Another use is in research to just look at how cells are behaving. So they can be made to identify specific molecules in a cell. So here you can see in that fluorescent diagram, you've got monoclonal antibodies with a fluorescent dye attached. So you can see where it's attaching to and where those antigens are within cells or within tissue samples. So we can learn more about how the cell functions. And the fourth use is you might want to use it to treat some diseases. For example, cancers are now being treated with monoclonal antibodies, where the antibody will attach to the antigen on a cancer cell. And instead of having some sort of fluorescence attached to the antibody, you might have a drug that targets that cell and will cause the cell to stop growing. So it delivers the drug directly to the cells. It doesn't harm other cells that don't have the antigen. So it's a very much a targeted way to deliver drugs to cells. And there's a bit more information on there if you want to stop and have a little read. Monoclonal antibodies are also used to detect plant diseases. So we'll have a look at that in just a second, but they can be used when you see a plant with a disease and that plant might be stunted in growth or have spots or decay or malformed leaves or different growths in different parts, or their leaves or parts of the plant are discoloured, or there might be a presence of press. They're always spotting if a plant has disease. And sometimes it's easy to work out what type of disease they've got, sometimes it isn't. So you could see, like, if it was black spots, it might be rose black spot or tobacco mosaic virus, which you should know from the combined content. These are black aphids, so that's a pest on this plant. And this is discoloration of the leaves. So it's kind of harder to work out when it's discoloration like that, because it might be something like a nutrient deficiency, or it could be a specific type of pathogen or something else. So the best way to identify a plant disease is to refer to a gardening manual or website to have a look and see if you can identify it that way. Or you could take infected plants to the lab to identify a pathogen or send off your sample. Or again, here comes the monoclonal antibodies. Use testing kits with monoclonal antibodies so that they can look for specific pathogens, for example, that are present in your plant. So as we said, when a plant shows its disease, it might have black spots or discoloration, but there might be different things that are responsible for it. So one of the things could be pathogens. So that's when you've got a viral or bacterial or fungal infection. And you can see that rose black spot there, you can see the black spots on there, and tobacco mosaic virus are two of those pathogens that you should know about from the combined content. These pests, those aphids that were shown before, this is a green aphid. What they do is they attach their proboscis into the phloem of plants and they remove the sugars. So it directly goes into the aphid just by going into the phloem. And therefore, if a plant has fewer sugars, then there will be less proteins that they can synthesize from that and less growth. So aphids are a problem for plant growth. And there's also nutrient diseases. So plants need to absorb nutrients from the soil. And if they don't absorb the correct amount of nutrients, then they won't be able to synthesize bigger molecules, just like proteins require nitrates, and DNA that will also require nitrates, and other nutrients will be needed to make other molecules for the plant to grow. The nutrient deficient diseases are quite important because their mechanisms for absorbing these ions is always through active transport in the roots. So they've obviously got to have enough nutrients in the soil to be able to do this, and also be able to have enough energy to be released from respiration for active transport to occur. So nutrient deficiency diseases can be quite common, but they may have different reasons for them happening. So a nitrate deficiency, if you don't have nitrates, then you cannot synthesize amino acids, therefore you can't make proteins. And if there's fewer proteins, there will be less growth. So one of the symptoms of nitrate deficiency tends to be smaller growth of plants. If you've got a magnesium deficiency, though, the magnesium ions are needed to make chlorophyll. And chlorophyll is the green pigment that captures light for photosynthesis to happen in chloroplasts. So if there's less chlorophyll, you get this chlorosis, which is a discoloration of the leaves where they start to lose the green pigmentation. If there's less chlorophyll, there is less photosynthesis, less glucose made, and glucose is the monomer that's used to make all of those different molecules for growth. So you will have less proteins and less growth. So not only will you see chlorosis on the leaves, you will also see less growth of the plant. It might be that you're asked to apply some of this knowledge in an investigation. This is actually a required practical that more lends itself to paper 2. It's not a required practical on paper 1, but it doesn't mean to say that you wouldn't get some sort of application question about some of these ideas. So you can imagine a scenario where you've got germinating seedlings growing on just distilled water, or just with some magnesium ions, or just nitrate ions, or magnesium and nitrate ions. In that scenario, you are changing the type of mineral ion in the growing solution. So that would be your independent variable. Your dependent variable, you could have a number of different ways of measuring this. For example, you could use the length of the seedlings, but you could also measure their biomass, which remember is the dried mass of the seedlings. So you'd remove the water, dry them out, and then just measure their actual dry mass. So that would tell you how much growth had happened in those seedlings. And if you're thinking about what controls could be in such an investigation, it would be like number of seedlings in each dish, the temperature they're grown at, the light intensity, the concentration of the different ions that they're going to use, and how long you're going to leave them to grow for, and the fact that you're measuring the dried mass, so making sure that they were completely dried out seedlings and there was no water left in them. So that's a sort of investigation you could do to look at these nitrates and other ions in a sort of an investigative way that could be an application question of those ideas. Plants also have their own defense systems. They don't have an immune system. So you shouldn't be talking about white blood cells or anything to do with plants. So their defense systems basically rely on physical, chemical, and mechanical adaptations. When you think of a physical defense, you're trying to stop any microorganisms or other pests from invading them. So things like thick bark on the trees, which is layers of dead cells, then that would prevent some animals from getting into the phloem inside. The tough waxy cuticle and strong cellulose cell walls, they're all mechanisms of defense to reduce how well they are going to get predated upon. The chemical plant defense responses are things like the actual chemicals that they make. So antibacterial chemicals are quite common, and some of those plants on the right-hand side have got some antibacterial chemicals in them. They might have poisons to deter herbivores. Different plants will have different poisons to different organisms, so they are just chemicals that they can use to stop other organisms eating them. And then mechanical adaptations, you're really thinking about the thorns and the hairs. So there's some like the nettle stings there, and leaves which droop or curl when touched, and also mimicry. So they can try to have leaves that might trick animals and prevent them from being eaten. Another bit of content you need to know for the triple paper, and the last bit of content is about bacterial growth. Now, there is a required practical associated with this, but again, you could be asked quite a challenging question about how do bacteria grow. So bacteria need to be grown in aseptic conditions on nutrient agar plates. Remember, bacteria have one circular chromosome, so they cannot divide by mitosis. They instead divide by the process of binary fission. And they divide so rapidly that if you are going to look at how quickly they grow, you have to use a logarithmic scale. And the reason you use this scale is because the range of numbers is so large. We start from like 1 or 2 bacteria to basically millions. So you are going to have a large range, and the only way to fit that growth onto a graph is to use a logarithmic scale. So here's a log graph of the growth of bacteria, and you can see that all bacteria on dishes grow in this way. So you can see initially you've got a lag phase where you've got no change in number, well, no real visible change in number, because they are divided by binary fission, but they're in such low numbers that it's difficult to see that change on a graph. Then you get the log phase of growth, which is basically your exponential growth, where they are multiplying at a constant rate in quite a rapid rate as well. Eventually, they start to run out of nutrients, and they also produce toxic chemicals. So the number of bacteria being produced is the same as the number dying. So there's no overall growth when you get to this last phase at the top, which we call the stationary phase. And then at the end, the numbers start to decrease because there's more toxins and not enough nutrients. So there's more death. We call this death phase. So those phases, you don't need to know the specific terms for them, but there have been questions in the past where you've been asked to describe what's happening at each stage. So it's worth sort of understanding how these growth phases are shown on graphs. But the real thing with the bacterial growth is to do with aseptic techniques, and there is a required practical. And for this required practical, you might be asked to be given a method or interpret results. So what you have to have is a general understanding of how we use aseptic techniques. So most of the solutions that you will use will be things like nutrient agar. That starts off as a liquid solution because you heat it up and then you leave it to set to make your agar plate. That must be sterilized. So any solutions must be sterilized. And the way you sterilize things is to use an autoclave. And this autoclave will heat solutions that you're going to use up to and beyond 120 degrees C, which will kill microorganisms. Once you've made your nutrient agar and you've sterilized it in the autoclave, you can pour them into a sterile agar plate. These are usually plastic Petri dishes, and you pour the agar on, and you keep the lid on, and you keep them sealed until you're ready to use them. When you are ready to use them, you're going to inoculate them with some bacteria. Now, there's two ways you can do this. You would use an inoculating loop. And to sterilize the loop, which is made of metal, you would hold it into a Bunsen burner flame and it will glow red. Then you leave it to cool nearby the flame, dip it into your bacterial culture, and you would perform a streak plate. So you would start by streaking in zigzags, and then you would streak in different directions, and eventually you end up with individual colonies. So you'd use inoculating loops to end up with colonies. If you want to make a lawn of bacteria, like this agar plate at the bottom shows, a lawn is growth all over the plate. You'd have to use a spreader. You can just about make out the glass spreader here in that shape there. It's sort of an L shape. And this spreader is made of glass, so you can't just hold it in a Bunsen flame. You have to dip it in ethanol and then set fire to the ethanol in the Bunsen flame. Not by holding it, just by letting it on fire. That kills all the microorganisms that might be on the spreader. So then you would use a sort of sterile syringe to put a drop of your bacterial culture on there and spread it around with the spreader. While working with any of these, you would have a Bunsen burner lit, and that Bunsen burner helps to keep the environment around you sterile. You need to shut all the windows so there's not loads of air moving through. Place your Bunsen burner on a yellow flame and just work nearby. That will kill all the microorganisms in the air, so it shouldn't land on your agar plate. With these as well, using good aseptic technique, if you're using a bottle, you should pass the bottleneck through a Bunsen flame. That would kill all of the bacteria. Sorry, it should be a blue flame, not a yellow flame. It should be the hot flame. You pass the bottle through the Bunsen burner to kill any microorganisms around the neck of the bottle. If you're using the nutrient agar and pouring it into the plate to leave them set, they're obviously going to be hot because it's liquid and it's going to cool and condense. So you should store your agar plates upside down so that the condensation doesn't drip onto the agar plates. These aren't upside down at the minute. They should be flipped so the lid is at the bottom. And that makes sure that you're reducing the chance of any microorganisms you don't want growing on your plate. The plates are sealed in tape when you've inoculated them, just in 4 places. This secures the lid on tight, but it doesn't it still lets oxygen in, so it stops any harmful bacteria from growing who might prefer anaerobic conditions. The plate should also be labelled on the agar side, which is the bottom of the plate, because the lid can move around. So this is to make sure that you can identify like whatever you've put in each section clearly, because the lid might spin round, whereas the agar will not move. The plates, when you've inoculated them and take them up, but they're going to be grown in an incubator at 25 degrees C. This is lower than body temperature to reduce the chance of human pathogens growing. This is what we use in schools. In hospitals and places, they would use warmer incubators because they don't have the same sort of preventative risk assessments that we have in schools. But 25 degrees C helps to prevent harmful bacteria growing that could cause you harm. And this really is your method. If you're going to use all those aseptic techniques, this is really what you do. You would inoculate the plate with the sterile syringe with the bacterial culture. Then you would get your sterile spreader and you would spread the bacteria around to create that lawn of bacteria. And the kind of practical you'd normally do is to do with antibiotic or antiseptics. So you would get some filter paper discs, which are sterile, and you'd soak them in a variety of antibiotic or antiseptic solutions, which should also be sterile. And you can use different solutions or different concentrations of different antibiotics. It depends what your independent variable is going to be. One of the discs should always be soaked in sterile distilled water to act as a control. This is not a control variable. This is called a control. And this type of control helps us to compare our different antibiotics or antiseptics to just a standard control of distilled water. If you use sterile tweezers, then you're going to make sure that you're not introducing unwanted bacteria, and you only lift the plate nearby your Bunsen burner when you're working. And as soon as you've inoculated your plate and placed on your discs, then you put the lid back on and you tape it in 4 different areas so that you allow the oxygen in, but don't get any harmful anaerobic bacteria growing. And then normally would incubate for around 24 hours at a maximum temperature of 25 degrees C. When you get your agar plates out after 24 hours, you would have thousands of colonies, and that's what makes it look like a lawn. Each little, the white stuff there is just individual colonies. Each of these colonies contains the clones of one type of bacteria. So the antiseptic discs, when you put them on that, what they're basically doing is, if you do not see the colonies growing, if you do not see that lawn growing, is the antiseptic disc has diffused through the agar and killed the bacteria in that area. And we call that area where there is no bacterial growth a clear zone or zone of inhibition. And this clear zone is basically where bacteria have been killed. If there are no clear zones around your disc, then these bacteria are resistant to that antibiotic that you have tried to use on your disc that you've placed there. So if you want to interpret the actual results and see, for example, which antibiotic might be the most effective against the bacteria that you've grown, you would need to measure the diameter of the clear zone. That's the zone around the disc. And then work out the radius from that, and then use pi r squared to work out the area of colonies that have been killed. And then you can look and compare the different size of the cross-sectional areas around each disc and work out which antibiotic has been the most effective. You might see on there some antibiotic resistance. So if you've got a clear zone with 1 or 2 colonies in there, then those colonies will contain antibiotic resistant bacteria. Some bacteria have a mutation in their DNA which can give them resistance to different antibiotics. This is a random mutation that they have, and this mutation will help them to survive and reproduce. So you can see in the diagrams at the top, you've got the green bacteria there, and they are going to be the sensitive strain. And then you've got one with a random mutation, which is the pink one, which is resistant to the antibiotic. Those sensitive bacteria will be killed, but the one bacteria that is allowed to survive, has got that mutation, can survive and reproduce by binary fission, and therefore you have now got a resistant strain of antibiotics. There are more bacteria within the mutation that survive, and they are not going to be killed by that particular antibiotic. So at that point, you need to try to use different antibiotics in order to kill them. And they've also got the advantage of there's less competition from other bacteria that were killed by the antibiotics, so they are able to grow quite rapidly. So this is leading us to antibiotic resistance. And there's more about that in the Combined Science B3 unit. Okay, that's all you're going to need for your triple content. I hope that's helped. Make sure you go back and have a look through anything if you need some extra help, and good luck with any exams.